Hi, Professor Runlin here. Let's take a look at antibiotics, antibiotic resistance, and detection of resistance in the laboratory. Nowadays, when you get a bacterial infection, you head to your doctor for a prescription of antibiotics. But that was not always the case. Prior to the mid-1900s, infections were the leading cause of death for human beings. Three huge advances, namely sanitation practices, including safe drinking water supplies and sewer systems, vaccination programs, and antibiotics, have changed all that. Thanks to these and some other changes, we are now expected to live 30 years longer than if we were born 100 years ago. Today, we are much more likely to die of cardiovascular disease or cancer than some little old infection. If you were to Google discovery of antibiotics, the search results will likely take you to the early 1900s, but antibiotics have actually been used by healers for a millennia. According to a written papyri 5,000 years old, Egyptians, for example, applied moldy bread to infected wounds, and the Ottomans in the medieval times took moldy cheese into battle campaigns to treat their wounded soldiers. And in the late 1700s, James McKeer, a North Ireland parish priest, used the local soil to treat infections. None of them knew why this worked sometimes, but it did. After the invention of the microscope, when we could actually see bacteria, and then the development of germ theory, that is, the idea that a pathogenic microbe causes an infectious disease, we could understand infections much better and could start to isolate and experiment with antibacterial compounds. So, when you Googled discovery of antibiotic, it most likely brought you to either Paul Ehrlich with the discovery of the chemotherapy agent salvarsan to treat syphilis, or Alexander Fleming with the discovery of the antibiotic penicillin. Confused? Well, let's look at a definition and then an explanation. Technically, a chemotherapy agent is a synthetic compound used to treat disease, including, but not limited to, infectious diseases, whereas an antibiotic is a biologically produced compound that is toxic to microbes. Many antibiotics are produced by fungi, which is why that moldy bread and moldy cheese and soil sometimes worked for treating bacterial infections. The distinction between chemotherapy agents and antibiotics, those terms, have become blurred because many of our newer antibiotics are actually chemically modified biological products or even chemically synthesized biological products. The discovery of penicillin started the golden age of antibiotic discovery, which peaked in the 1950s. Half of the most commonly used antibiotics today were discovered between 1950 and 1960. Antibiotics are toxic to bacteria because they interrupt cell structures or cell processes. For example, all of the penicillin-related antibiotics prevent the formation of the cell walls. Tetracycline disrupts protein synthesis, and the sulfa drugs interfere with the biochemical pathway. Because antibiotics target different bacterial structures or functions, different antibiotics work better against different types of bacteria. I wish I could report that antibiotics have cured all of the infectious diseases, but sadly, they have not. In any ecosystem, the organisms are constantly evolving and adapting, and this is no exception. As we developed and used more antibiotics, the bacteria developed antibiotic resistance, ways to avoid the toxic effects of antibiotics. In fact, in 1941, the first human received penicillin, but even before that, in 1940, during lab experiments, it was found that E. coli could make an enzyme that effectively inactivated penicillin. And by 1942, it was well established that Staphylococcus aureus seemed to spread the resistance to penicillin from one bacterium to the next. In addition to penicillinase, many other resistance mechanisms, such as antibiotics efflux pumps, Altered metabolic pathways and target modifications have been identified. Research experts believe that resistance is promoted by the overuse of antibiotics, agricultural uses, and patient noncompliance. Today, antibiotic resistance is considered by some to be a worldwide crisis. Certainly, the reports of bacterial species that are resistant to all known antibiotics leave us very vulnerable to common infections. Now that we have a little background, you can see why it's essential that we develop laboratory techniques to detect antibiotic resistance. After all, if a patient needs to be treated for a Staphylococcus aureus infection, to provide effective treatment, you would need to know if the bacteria causing the infection are resistant to certain antibiotics. And in the mid-1950s, researchers proposed a standardized disc method. And in the early 1960s, this method, the Kirby-Bauer disc diffusion susceptibility test, became the accepted standard. The basic steps are: distribute the bacteria evenly across the surface of an agar plate, dispense discs impregnated with the antibiotics on the plate, incubate them, and then examine them to see if the bacteria can grow near that antibiotic or not. Ready? Let's get into the details and see that procedure. To start with, you will need a properly prepared Mueller-Hinton agar plate and a standardized broth culture of bacteria. During our lab, we typically have each student make 4 plates, one for Staphylococcus aureus, a gram-positive bacteria, one for E. coli, a gram-negative bacteria, one for Pseudomonas, a notoriously antibiotic-resistant gram-negative bacteria, and one for Mycobacterium smegmatis, an acid-fast bacteria. Start by labeling one plate for each bacteria. When inoculating these plates, you need to spread the bacteria across the entire surface of the agar. To do this, shake the tube and open it using your aseptic technique. Then dip a sterile swab into the inoculum. Rotate that swab against the side of the tube just above the fluid level to remove any excess fluid. You don't want it dripping wet. Inoculate the surface of the plate by streaking that swab across the entire surface in one direction. Then rotate the plate about 60 degrees and streak across the entire surface again. Rotate it one more time, swab that whole surface from the new angle. Then discard the swab in the biohazard bag at your pod. Leaving the lid slightly ajar, allow the plate to just sit there at room temperature for 5 minutes. In our lab, we use an antibiotic dispenser to place those antibiotic impregnated discs with the known concentration of the antimicrobial compound on the surface of that agar. To do this, simply remove the lid from the plate, place the dispenser over that plate, and firmly press the plunger down just once to dispense the discs onto that surface. Then lift the dispenser off the plate. Use an alcohol pad to wipe the bottom of the dispenser. Note that each disc has letters on it. This indicates the name of the antibiotic on that disc. When those discs are placed on the plate, immediately water is absorbed into the disc from the agar, and that antimicrobial agent begins to diffuse into the surrounding agar. To ensure that the discs are making solid contact with the agar, dip the tips of a forceps in alcohol and quickly flame the tips. Then touch each disc on the plate. Incubate the plates for 24 hours at about 35 degrees Celsius. Note that after incubation, some discs will have clear zones around them and some discs will not. The clear zone is referred to as a zone of inhibition, and it indicates that the bacteria growth was inhibited by the antibiotic. The presence of inhibition is not enough to determine resistance. You must measure the zone and then use established standards to make that determination. To do this, view the bottom of the plate and measure and record the diameter of the zone of inhibition for each disc. Note that the measurements must be in millimeters. If two zones overlap or the disc is close to the edge and you can't quite measure across the entire diameter, find an edge of the zone, measure to the center of the disc, and then multiply by 2 to get the diameter. If there is no zone, that means there's growth right up to the edge of the disc, and report it as 0. Sometimes the results aren't quite as clear-cut. You may see two zones, a smaller one very distinct and clear, and then a larger one a little less distinct. Measure only up to the distinctly clear zone lines. If there is a zone of inhibition that has colonies inside the zone, those bacteria may be resistant to that drug. You can only measure the diameter of the area with no colonies. Once all the zones have been measured, you will need to interpret the results using the current guidelines established by the Clinical Laboratory Standards Institute. Using their chart, find the antibiotic you tested for and the bacterial group. Then compare the size of the zones of inhibition to determine whether the bacteria is R, resistant, I, intermediate, or S, sensitive. For example, if I was testing Staphylococcus species for penicillin resistance and the zone was 23 millimeters, the chart indicates that that Staphylococcus species is resistant to that concentration of penicillin. In other words, the concentration of penicillin tested is not a good choice for treating that infection because the infecting bacteria are resistant to it. The treatment needs to be with an antibiotic that the bacteria is sensitive to. If the bacteria are sensitive to the antibiotic, it means that the antibiotic will kill the bacteria. Since bacterial infections are still common and antibiotic resistance is increasing, this lab technique is really kind of important. In the video description notes, I have included a website link that allows you to quickly see resistance searching by either the bacteria or the antibiotic or a specific country. I hope you like this lab. Work safe.